Hello, we are going to start talking about the histology, or microanatomy if you prefer, of the musculoskeletal system by looking at the nerves of the peripheral nervous system and how they actually are getting to the skeletal muscle, and then the features of the skeletal muscle tissue itself that allows it to do its job. So to do this, we're going to start by going down and finding a section of peripheral nerve right here. We'll open that up. And this is going to be a cross-section. So the cross-section is going to show the nerve as it was cut kind of transversely. So here we see a cross-section of the peripheral nerve. I'll just kind of get things pretty here so we can see what we're looking at. And so here, what we can see is that the nerve has a large sheet of connective tissue covering it. This is called the epineurium. Epi for above or outside. So epineurium is the connective tissue collagen that's surrounding it. And each nerve, the peripheral nerve here, is made of irregularly shaped fascicles. These are collections of axons and their support cells. Now these are surrounded by connective tissue that's called the perineurium. So epineurium on the outside, perineurium around each individual fascicle, and surrounding each individual axon and support cell inside of the fascicles is what's called endoneurium. Now that's a little bit tough to see in this preparation. We can't really see the endoneurium as well in this hematoxylin and eosin stain, but what we can see as we zoom in very close are lots of nuclei. These darker purple structures here are the nuclei of the support cells of the axon. These are called Schwann cells. So the Schwann cell nuclei are here, and these cells support the axons. Here we can see an axon. I'm just hovering around it with the cursor. And that axon is going to have what's referred to as a myelin sheath around it that's produced by the Schwann cell. And then endoneurium is the connective tissue layer that surrounds each one of those constructs. And we can't really see the endoneurium too clearly here, but the individual axons are just these dots with some coloring in the middle, but not quite as dark as the nuclei of the Schwann cells. And that's going to be our clue that we've got the nerve here. Now, since we're in the peripheral nerve, we don't actually have any nerve cell bodies present. Those are going to be in special places like the spinal cord or ganglia. And because nervous tissue is just like anything else, it needs nutrients, so we're always going to find blood vessels, maybe small little capillaries, getting close to capillary size here, larger vessels, and sometimes even quite large vessels, like this nice big artery here with its multiple layers of smooth muscle just outside the lumen there. And here, now that we've seen all that, we'll just do a quick little view here of the labeled structures. Now one, see if you can identify it. That's the connective tissue surrounding the whole nerve. That's the epineurium. And I'll give you a moment here with these smaller scale structures. Try to zoom in, but not actually leave anything unseen. Take a second here. Alright, so what we're seeing is this kind of light blue arrows pointing at the perineurium. 3, these lighter yellow arrows are pointing at the nuclei of the Schwann cells. And 4 are pointing at these kind of indistinct, slightly paler blobs. And these are the individual axons at the tip of the darker blue arrows surrounded by their myelin sheath. Let's take a look at another section here. This is another cross-section through a peripheral nerve. And here we can see that this cross-section through the peripheral nerve looks much different than the last one. That's because this is a different stain. This is a Mason stain, and we've got, instead of the H&E, purple and pink, we've got connective tissue showing up this nice big kind of layer of turquoise blue here. So that's what makes this distinct. Now the other thing that's important about this stain is that it's giving us a nice view of the myelin sheath and the interior of the cell. So if I look right here, we can see this circle with the bullseye right in the middle. The bullseye is actually an axon. And the surrounding tissue is, or the surrounding area, pardon me, is where we have the myelin sheath laid down by the Schwann cell. Now, some stains are good at showing some things and not others. This stain doesn't really show us the Schwann cell nuclei. So we can see the boundary of the Schwann cell and the outer rim of it, which would be that endoneurium, we can see the axon, but we don't really see the Schwann cells themselves. Some of these you can see primarily in this section. So now, let's zoom over to this other smaller fascicle here and see if you can identify the structures that are indicated. So here, number 1, this turquoise arrow is pointing towards this turquoise blue. This is the epineurium, but since it's surrounding a single fascicle, you could also make a case that it's perineurium. So I wouldn't get too particular about this one, epi versus perineurium. We do have some blood vessels here, as always. 2, this is pointing at that space within the myelin sheath. So that's the space within the myelin sheath that surrounds it. And then here, the purple arrows are pointing at the outer boundary. So that outer boundary is where the limit of the Schwann cells are and where we would find the endoneurium. Now, one thing that's not labeled here are the axons, but we can actually see lots of those. Any of these bullseyes in the middle of those clear areas would be examples of axons. All right, we're going to take one more look at a cross-section of a peripheral nerve before we move on to a slightly different view. And in this case, we're looking at it in a really unique way. This is an osmium tetroxide stain. And as we mentioned before, some stains are good at showing some things, but not others. This stain is incredibly good at showing us where the myelin sheaths are. In fact, all the black stuff is the osmium tetroxides filling the space that would have been filled by the myelin. So all of these tires that we see here, those individual round circles with a clear center, the dark area is showing us where the myelin sheath is, and the area in the center that's kind of clear is where the axon would be. Now if I look at this one just here, and you look in the bottom right corner, you can see a slightly magnified area. You can actually see what remains of the axon right in the center, although it's not as prominent in some of the others. So we can see connective tissue here, but this stain is not one we'd use to see the details of the connective tissue, but the myelin is showing up nice and clear. So let's see if we can do a quick little quiz, and you can tell me what you see here, arrow number 1 and arrow number 2. All right, well, of the things that we've been looking at today, arrow number 1 is pointing at the myelin sheath, and 2 is pointing to that inner area which would contain the axon. We're now going to switch gears slightly and look at the peripheral nerve, not in cross-section, but in a longitudinal section. And this longitudinal section, basically, if we were to qualitatively describe it, looks like a bunch of wavy lines. But what's cool about this is that these wavy lines, even though they look similar, are going to be showing us connective tissue and nervous structures of this peripheral nerve in its longitudinal state. So if we kind of zoom in here, the outermost layer, this is the epineurium, and it's made of collagen. So type 1 collagen, which means that any of the nuclei that we see in here, these long spindly guys, these are fibroblasts. Now just over from that, we come to nervous tissue. And I'm going to zoom out again, and we'll pick up right here, because this is a nice illustration, that nervous tissue tends to have this undulating appearance in longitudinal section. Basically, it's got this wavy appearance. It's kind of smooth. So this is what a nerve fascicle looks like. So we've got the epineurium on the outside, but then we've got connective tissue here separating this fascicle from its neighbor. So this is going to be perineurium. I'm going to zoom in here, and the perineurium and the nearby nervous tissue, they look similar. But remember that nerve has this smoothly undulating character, whereas the collagen bundles of the perineurium looks more like kind of slightly unraveled yarn. It's a little more chaotic looking. So these individual collagen bundles here are these large pink structures with their supporting fibroblasts, whereas over here we have the peripheral nerves, the axons, surrounded by their many Schwann cells. And there are a ton of Schwann cell nuclei throughout this area because each Schwann cell forms a myelin sheath or supports an area of an axon, just a small bit of it. So each axon has many, many Schwann cells up and down its length, and each of its neighboring axons also has many Schwann cells up and down its length. So these nuclei in the nervous area, these are all Schwann cells. Now I'm going to zoom out and come back over here because I found a nice example of this. Let's see if we can find it again. Very good. So right here, you can look in the bottom right corner for the zoomed in image if you like, but right here we actually can see a thin stripe, a little thread right at the core of this myelin sheath. That is the individual axon. And the myelin sheath contains a lot of fatty material, and fatty material is removed during staining, but sometimes leaves behind this foamy appearance. So this foamy looking stuff, right where I'm moving the cursor, is the remnants of the myelin sheath, and that little thread right in the center of it, that is the axon at the core of that sheath. We're going to now zoom back out. Hopefully no one's getting motion sick. And we'll zoom in on the self-quiz area here. And you can take a moment and see if you can figure out what's what. Alright, so we've got 1 pointing at this structure and 2 pointing at this structure. So 2, this is that smoothly undulating nervous tissue, whereas we've got the unraveled yarn here of the perineurium. So 1 is perineurium, 2 is the nerve fascicle. 3 is pointing at nuclei, and we don't have any nerve cell nuclei in peripheral nerves. Instead, we've got the support cell nucleus. These are all Schwann cells. And here, 4 is pointing at that little thread in the middle of that foamy remnant of the myelin sheath. 4 is pointing at the axon itself. Our next slide is also a longitudinal section of a peripheral nerve, but we have a different stain. This is that Mason stain, once again, which we can sometimes spot because of that turquoise blue that it gives to some of the connective tissue collagen bundles. But what's really nice about this slide is, as we mentioned earlier, it doesn't really show nuclei well, so we don't really see all those Schwann cell nuclei that we saw in the H&E stain. But what we do see is the outline and cell boundaries around those individual myelin-producing Schwann cells. So here we can see the axon moving from one Schwann cell, boom, to another. And the myelin sheath for one stops here and another picks up right there. So this is where two Schwann cells meet, and that meeting point is known as a node of Ranvier. And that node of Ranvier is where a lot of surface proteins are clustered that will allow the what's referred to as jumping or saltatory conduction of the action potential down this peripheral nerve. So that's the node of Ranvier right there, and the more we look for them, the more we see. They're there. Let's just scan randomly. We'll see more. Right there, and that little thread that's traveling between those adjacent Schwann cells, that is the axon itself. So let's come on over here, look at our little self-quiz area. Well, first off, let's look at number 1. Number 1 is pointing at that outer layer. Now this is a little bit dicey in that it's the outermost layer, so we want to call it the epineurium, but it's also surrounding an individual fascicle, so we could also make a case for it being the perineurium. So we're not going to get too hung up on that, and it would not be something I personally would test you on, that you have to tell if this is peri or epi. But since it's outermost, calling it the epineurium is a fairly safe bet. But let's get down to something a little more concrete. So what do we see at 2 and 3? Two is pointing at the junction where two Schwann cells meet, so that is a node of Ranvier, whereas 3 is pointing at the individual thread that's traveling between those through the node, and that is the axon itself. We're now going to switch gears rather starkly, and instead of looking at a peripheral nerve, we're going to look at the actual cell bodies that are going to contribute to part of that peripheral nerve. This is what's known as a posterior root ganglia, also commonly known as a dorsal root ganglia, and it is a place where we find many neuron cell bodies, and in this case they are sensory cell bodies. So these large blobs right here, these are the large neuron cell bodies. So we'll just zoom in on one at random. That's a good example. I think this guy is a nice one. Let's see if there's another. Okay, that one's even better. So here we go. This large structure here, this is the cytoplasm and nucleus of a neuron cell body. Now, a neuron cell body is also sometimes called the soma, so you can also kind of file that away. But these sensory neurons are kind of special in that they have a single axon that splits and goes to the periphery, and then also splits and goes towards the central nervous system, the spinal cord. And so we don't really have any dendrites sticking out of here. So we tend to have a fairly even coating of cells around it. These are going to be the support cells of the neuron cell body, and these are called satellite cells. So the nuclei of these satellite cells are these little kind of darker bulbs right here that we can see surrounding it. Now if we look around a little bit, I can probably find one that's almost completely surrounded. So if we look at this, there we go. If we look at this one right here, we can kind of see how it's surrounded almost uniformly. Oh, here's an even better one right here by these satellite cells. That's a particular feature of these posterior or dorsal root ganglia. Other nerve cells have support cells, but because they have dendrites sticking off at various angles, they're not going to be as evenly coating. Now to zoom back down here, you can see that the nucleus has a darker area inside of it. This is going to be the nucleolus. The nucleolus, having that large dense area, means that there's also areas of DNA that are unraveled that's making it transcriptionally and translationally active. So this is a nerve cell that's producing a lot of protein, or in this case, likely neurotransmitters that are going down the axon to let the neuron do its job. And if we zoom out, this H&E section isn't great at showing the axons, but that's what we have pretty much all the way through here. Now I say that, and then I immediately find a really nice looking axon. So right here, we have an axon surrounded by the myelin sheath put together by its Schwann cells, likely nuclei of Schwann cells here, and here, and here. And these axons will be leading to a ganglion and then projecting to the central nervous system. So that's pretty much all the features that I wanted to show you here. Let's zoom in on this area, and you can just take a moment or two and do a self-quiz on what you think each of these structures are. So number 1, these green arrows, they are pointing at the satellite cells. 2, the blue arrows are pointing at the nucleus, and 3 is pointing at the nucleolus. Within the nucleus, that dense area, showing that we've got transcriptionally active nuclei here. Now that we've seen the nerve cell bodies inside that posterior root ganglia, we're going to take a look at nerve cell bodies in a slightly different place. These are parasympathetic ganglia. Now ganglion typically just means a collection of nerve cell bodies outside of the central nervous system, so outside of the brain stem, or the spinal cord. And now most of what we're looking at here is the prostate gland. But the prostate gland, being an organ, has to have innervation, and nerve cell bodies clustered in areas around their wall. Now we've got the labeled area here. I'm just going to see real quick if I can spot some other examples of those ganglia, and then we can revisit that. And here we go. You can see, just like in the posterior root ganglia, we've got these big soma here, these big nerve cell bodies, and we've got a nice looking nucleus and nucleolus there. And so these are surrounded by their satellite cells. But because these cells are not sensory, they have an axon and multiple dendrites coming in. So these satellite cells do not form that continuous layer of cells on their outside. So that's one way you can tell these autonomic nerve cells apart from the sensory nerve cells. The other thing is that they're in the wall of an organ, not in a posterior root ganglia. So you'll get better at picking up those cues as you go. We've actually got some nerve fascicles here leading to and from those, but we're not going to sweat that right now. So let's scooch back over here to this area where we can quiz ourselves. And I can zoom in here. Just tell me what you're seeing in arrows 1, 2, and 3. So arrow 1 is pointing at the nerve cell itself. 2, nucleus. 3, nucleolus. So like many nerve cells, this one is translationally active, and so it's going to be producing a lot of neurotransmitters and some other proteins. Now, it's hard to see the axons coming and going because the axons are very thin and the nerve cell bodies are very large. But if we look at this guy right here, if you see how it's forming a nice little spike at about 7 o'clock on the clock face here, that could very well be an axon. This could be the axon hillock forming the axon, but I cannot guarantee you that that's what it is. So we're not going to say definitively, but that's possible. And last but not least, we also have little satellite cells hanging out around the periphery of these individual neurons. And because I'm a sucker for completeness in these videos, I'm now going to take a look at the other kind of autonomic cell that we have, and this is a sympathetic ganglion. Now, sympathetic ganglia are going to have different effects than the parasympathetic nerves, and you'll learn more about that in various other classes and courses. But right here, we can see there's a lymph node right here, so don't get distracted by this large purple and pink structure. What we're looking at are the nerve cells that are nearby. Now, right here, we've got this consistently wavy structure here. So this is actually a nerve, likely going to this ganglia. So we got Schwann cells and axons inside there. But if we take a second and look, I don't really have too many other to choose from. So I am going to stick with the one right here, and is a cluster of nerve cells. We can see once again the nerve cell body here, it's soma. We've got some satellite cells around it and the nucleus and nucleolus right there. One thing I want you to note here that we can see a little more nicely than some of the others up until now is that many nerve cells accumulate these brownish little kind of inclusions inside their cytoplasm as they age. This is called lipofuscin, and it's a very common thing to see. It's not necessarily pathologic, but because cells accumulate them as they age, they are associated with certain degenerative conditions, but possibly not in a causative way. So now let's jump back up here, and we can see the self-quizzing area. So just take a look at these arrows, and you can see here that the small red arrows associated with number 1, these are pointing to the very small cells that are nearby. So these could be these satellite cells around the neuron itself. But one thing that's possible is that these could also be some of the Schwann cells that are supporting the axons that are coming and going from these cell nuclei. 2 is pointing at the nerve cell of, in and of itself, whereas 3, nucleus, 4, nucleolus, are present right there. We're now going to switch gears just a little bit and take a look at the spinal cord and some of the features that are there that are similar to what we've seen before, but have their own little twists. So first off, one thing you might note is that the spinal cord is upside down compared to the way that you usually look at it in books. So we've got the posterior side here, anterior side here. So the anterior ventral horns are here and the posterior horns are here. And if we zoom in on the gray matter, that's where we're going to see the nerve cell bodies. So we've got those here. We can see the nucleus and nucleolus right there. A lot of support cells, satellite cells nearby. And there's not much of a self-quiz here, so we'll just zoom in real quick. And we've got this nerve cell here in the intermediolateral cell column. It's going to be one of the sympathetics, but we've got a nice view of the axon departing that cell right there. So if the gray matter cell bodies are on the inside, internal part of the spinal cord, this outer part is going to be full of axons. This is white matter. And here we can see just axons everywhere. This little dot inside its sheath, here, 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 pretty much every little dot we see is going to be an axon surrounded by its myelin sheath. But one thing we don't see are a great many other nuclei. And that's because unlike the peripheral nervous system, where the myelin sheaths are laid down by a whole succession of Schwann cells, in the central nervous system, of which the spinal cord is part, we have oligodendrocytes, and these cells send out multiple processes that myelinate multiple axons. So you don't need as many oligodendrocytes to myelinate the same number of axons as you do with the Schwann cells in the peripheral nervous system. So that's one reason we don't see a whole lot of other nuclei here, and that the axons are much more prominent and appear to be much more densely packed. All right. Just before we're done here is a quick little fun part of the tour. We've got the outer layer here, the pia mater. We've got the cobwebby arachnoid mater with the vessels going to the spinal cord, and we don't have the dura mater, but it would be another outer, thicker layer right there. And the nervous system to the muscles that it's innervating. And this is not on your self-guided tour, but we can see here a special silver stain that's been done, and it's showing us the nerves terminating on individual muscle cells. So we've got these muscle cells here. We can see their striations, but in particular here we can see the nerve end plates like this, where acetylcholine will be released and cause the muscle to contract. So we've got those highlighted here and here. Feel free to take a look at them if you like. That's really the only big feature of this is going to be that neuromuscular junction where the nerve meets the skeletal muscle. Alright, after all this to do, we are finally going to take a look at the histology of muscle itself. So we have three different types of muscle, and we've discussed this in a few different places, but here we've got an example on the left of smooth muscle. In the middle we have cardiac muscle, and here on the right we have skeletal muscle. Now, right now I'm teaching the musculoskeletal course, so skeletal muscle is mostly what we're concerned about, but there are features of smooth and cardiac muscle, that are going to be interesting as well. So let's start with smooth muscle. Smooth muscle in this case is surrounding the lumen of an organ. So we're looking here at, I believe, the gallbladder, and all of this is smooth muscle. Now remember that smooth muscle is something that we have non-voluntary control over. Our sympathetics, parasympathetics, and hormones can control the tone and contraction and relaxation in this muscle. And what we see, as a colleague of mine once described it, is little purple footballs in bubblegum. It doesn't have the same wavy appearance as peripheral nerve, it doesn't have striations of skeletal muscle, and it doesn't have the large collagen fibers that we would see in dense connective tissue. So here, this is a good example of smooth muscle, and we have these elongated smooth muscle nuclei present there. So if we take a look up here in the self-guided area, we can see, once again, the nuclei have been caught in cross-section here. We've got them a little more longitudinally over here. And that gives them that elongated appearance when they're in longitudinal cut. But you can notice that smooth muscle is taking multiple different courses through the wall of the gallbladder here. Now let's return to a little more macroscopic scale and take a quick look at cardiac muscle. Now again, it's not the main focus of what we're teaching right now, but cardiac muscle is also involuntary, but it is striated. So its method of contraction is somewhat similar to skeletal muscle, but we're not in charge of it. Hallmarks of cardiac muscle are we have these nuclei right here that are going to be a little more centrally located. They're inside the actual fibers of the muscle. We can see the striations here. We've got the dark and white lines, the A line, dark, the I line, light, present. But one thing cardiac muscle has that skeletal muscle doesn't are these dense areas here that mark where these cells fuse. Instead of nerves innervating every fiber, the contraction and depolarization of cardiac muscle passes from cell to cell. And in these dense areas, these cells are fused together, and a little less visibly along here we have gap junctions that allow cytoplasm to be shared between these cells so that a contraction or action potential can actually be passed from one cell to its neighbors. So we see these intercalated discs, centrally located nuclei, and branching of fibers in cardiac muscle. We do not see any of that in skeletal muscle. Here's a nice branching pattern right there. So that's going to be the hallmarks of cardiac muscle. So now let's take a look at the big show, skeletal muscle. This is actually taken from the tongue. We've got some taste buds, filiform papillae out here. We're not going to worry about them for right now. But essentially, skeletal muscle is voluntary. So it's going to be innervated by somatic nerves. It is striated, and it has peripherally located nuclei. So right here, we have a cross section. You can see how the nuclei are on the outside of these skeletal muscle cells. We zoom out a little bit and we'll find a longitudinal band, so hopefully somewhere besides where we already have it marked. There we go. And zooming in here, we can make out these striations. So once again, we've got the dark A bands, the light I bands, and these muscles, although they're running side by side, do not branch. They do not run into each other. They are separate along their entire length. There's no gap junctions. So unlike cardiac muscle, these cells remain separate. They do not pass their contraction from cell to cell, but have to be depolarized by nerves. So once again, we can see those striations in longitudinal section, and in cross section, we can make out the overwhelmingly peripheral nature of those nuclei. Now, one other thing I haven't brought a great deal of attention to is that we have myosatellite cells in skeletal muscle as well. They're going to be supporting these cells, but they can also differentiate to become full muscle cells as well and add their cytoplasm to this existing muscle cell. Skeletal muscle cells are multinucleated, so new cells will just fuse with it and add their cytoplasm to it and start laying down, creating new actin and myosin filaments that allow the muscle to do its job. Because muscle and its components are so important, we're going to hit some of those same features on another slide so that we can see how they are laid out and how those features work. Now, one thing I do want to note here that we didn't see on the prior slide is that muscle has connective tissue just like the nerves did. We have an outer lining of connective tissue. So we have collagen fibers and fibroblasts here. This is going to be called the epimysium. So epimysium is here, and then the individual fascicles here, which are a little bit less distinct than they were in the nerves, but the individual fascicles are separated by perimysium, and surrounding each individual muscle cell is a very, very thin layer of connective tissue called the endomysium. So that's not super obvious right here, but those differences are all present. We can see the real rough boundary of a fascicle here, so there'd be perimysium running along this area we see here. Here's a nice little fascicle separated right there, and each cell surrounded by endomysium. Because this is a muscle that's been cut in cross-section, we can't see the striations, but we can see the peripheral nuclei very, very easily. And the nucleus right there is just one of many that's associated with each muscle cell. Now this next slide contains both a cross-section and longitudinal section of skeletal muscle. Now, one thing that we can't really see in this section real easily, because it's a much bigger muscle, is the epimysium. We can't see the connective tissue on the entire outside of the muscle. However, with just a little bit of looking, we can see the perimysium separating individual fascicles of the muscle. So we've got the perimysium separating these fascicles and some connective tissue there. Also tends to be where we find nerves and vessels that are moving into the muscle. So here we have a peripheral nerve that's invaded the muscle to terminate on those actual muscle cells and cause them to contract. If we look closely here, a few of these nuclei have shrunk, but that gives us a chance to see the very, very slender endomysium. This little tiny strip right here is that connective tissue surrounding an individual muscle cell. So an individual fiber surrounded by that endomysium right there. Now if we take a look over here, we've actually got something very cool circled by number 5 here, and I'm showing it to you right now because we don't have any other examples in this slide. But basically we have some smaller isolated skeletal muscle cells surrounded by connective tissue within a larger muscle. This is one of the sensory structures that actually is inside muscle. This is called a muscle spindle fiber, and it senses the amount of stretch by this muscle and can adjust the tone of that muscle accordingly. We got a nice big perimecial layer of connective tissue right next to it, but this right here is a muscle spindle fiber. Now let's take a look at this cross section of muscle, and we'll zoom in somewhat at random here. We can see individual areas, fascicles separated by perimesium. In between each one of these cells would also be endomesium, but we're not really able to see that, at least not clearly, in this section. One thing we can see very clearly are the peripheral nuclei of these elongated muscle cells lining it on the outside, and if we zoom in we can see the striations, the actual stripes of these muscles right here. So we have dark A bands and light I bands. Light has an I in it, I bands are light, dark has an A in it. A bands are dark. Now if we zoom out a little bit, we can take a quick look at the self-quizzing module here, and we've got purple arrow right here. This is 1. Take a second, tell me what you think that might be. And that is perimesium separating nearby fascicles of muscle, groups of muscle cells. Over here, it's actually another. So you can satisfy yourself that that's a very similar thing. And right up here, 3 and 4. So number 3 is indicating the nuclei of these skeletal muscle cells, and 4 is indicating the cell itself, its cytoplasm packed full of striated filaments made up of actin and myosin. Now moving back over to our cross section here, let's take a look at the remaining areas and tell me what you think 2 and 3 are. 2, these lighter arrows are indicating the endomesium around each individual cell, whereas 3 is once again pointing to that peripheral skeletal muscle nucleus. Now here we have one last section of skeletal muscle, and we are beating this rather to death. However, this particular specimen, it's the tongue. We got the taste buds out here and papillae, but this one has the nicest striations of any that we've seen. Let me zoom in accordingly, make a little room here. So here we can just see the striated nature of the skeletal muscle cells so clearly. So right here, peripheral nuclei, A bands and I bands, super clear. Take a look at this specimen if you like. Notice we've also got adipose tissue nearby, and all these muscle fibers interdigitating with each other inside the tongue. In this slide, we're going to see how the connective tissue of the muscle contributes to the actual tendon that will move bones when the muscles contract. So here on the right side, we zoom in, and yet once again we see striations, peripheral nuclei, so we know we're looking at skeletal muscle. Each muscle fiber is surrounded by endomesium. Each fascicle or group is surrounded by perimesium. And the outermost layer of the muscle itself is this epimesium, this large pink connective tissue structure. And one reason it's so pronounced on this specimen is that this muscle is very close to the end where it's going to become completely a tendon and no more skeletal muscle tissue will exist. So we can see here that the skeletal muscle fibers are ending right here at the epimesium, and their connective tissue is fusing with it. And inside here we have thick collagen bundles and fibroblasts, these long stretched out nuclei here. No striations present in the collagen bundles, so that is not skeletal muscle, but nearby we can see it very clearly. And if we zoom up here, you can see the epimesium indicated by the green arrow, and a nice view of a strip of perimysium separating fascicles joining with the connective tissue there. And this is just going to keep collecting the connective tissue of this muscle as we get further and further away from its central area, and the muscle goes away, leaving only this dense, regular connective tissue behind, which will attach to bone as a tendon. Alright, from here we have one other quick aside before we're done. We've seen the muscles, we've seen the nerves, we've seen the tendons. Now we are going to take a look at something that's not in your self-guided tour, because it's really not something that you can spend a lot of time looking at. But here we have the jumble of muscle transitioning into tendon, and in addition to the connective tissue of the tendon, at that myotendinous juncture we sometimes find another sensory receptor. This is called a Golgi tendon organ. Now you would not need to find this in a perio. They're tough to find, but right here we've got a nerve terminating inside where the muscle and tendon meet, and that's going to help us sense stretch that is coming through that tendon and prevent excessive stretch that might avulse or otherwise damage it. So once again, Golgi tendon organs are one of those sensory structures we find right as the muscle becomes tendon.